Welcome to the beginning of chapter 28. We're going to do this in 3 chunks. One will be an introduction where we'll talk about mitosis, meiosis, and why we care. The second will be male anatomy specifically. The third will be female anatomy specifically. So let's go ahead and begin with the introduction. So here I have a cute little cartoon from Awkward Yeti as per usual, and our purple slides as usual. And here we begin with introduction reference slides. So first for this chunk, I want to talk about how to really best understand everything, because at first it'll sound like, oh, this makes sense, this makes sense, this makes sense. And then when you go back to try to review it for whatever reason, you'll be like, oh my God, I don't get it. Like why, like it made sense. Now all of a sudden everything sounds the same. I don't understand how this deals with this cycle and this deals with this other cycle or pathway. How do I keep it all straight in my head? So that's what I tried to do here with our lecture slides at the beginning. You see I have 3 points, microanatomy to macroanatomy. And I have a summary for both males and females that we'll talk about briefly, and then we'll go into the details about it later. And then the hypothalamic pituitary gonadal axis, so these are all the hormones, and then production of gametes. So for each male and female, there'll be more specific names for each of these like subsections that you'll want to add into these sets of slides, but for now let's just go ahead and intro them, so then that way whenever we start talking about them, you know where you can refer to it back in your notes. So each following slide goes into detail. So for the micro to macro anatomy, so going from the smallest scale to the largest scales, for males, we'll start with the seminiferous tubules, and then we'll end up in the urethra, and this is happening continuously. Things are moving continuously in this system. Versus females, we'll start in an egg nest and an ovary, then we'll move to the uterine lining, and then we'll have menses, and that'll go out through the vagina. Female cycles will include 3 different ones, but 2 referring to the anatomy specifically are the ovarian and uterine cycles. Ovarian is where we're actually making our eggs, and this is the follicular phase going into the luteal phase. Then we have the uterine cycle, and the uterine cycle is what's happening to the uterus. So the uterine cycle starts with the menstrual phase, then goes into the proliferative phase, then finishes out the secretory phase, and then it finally restarts back at the menstrual phase. When we're talking about the hypothalamic pituitary gonadal axis, we're talking from brain down. So in the brain, we'll start with the hypothalamus. For males, that'll start with GnRH. GnRH will talk to the anterior pituitary and tell the anterior pituitary to release FSH and LH. For the males, it'll go to the testes. The testes will start producing testosterone, which I abbreviate as T, a capital T. This starts at puberty. The frequency is always happening, and it's organized in pulses by GnRH, and then it ends during death. Of course, it'll taper off a little bit closer towards or in the elderly age, but for the most part, this won't finish until death. For females, same thing, except now we're going to the ovaries instead of the testes, and the ovaries are producing progesterone and estrogens. This starts at puberty, frequency is every 28 days, and it stops during menopause, typically. For productions of gametes, gametes are our sex cells. So for males, I have in parentheses S equals singular, and P equals plural. Plural? Oh yeah, that looks right. Okay. So for singular, we have spermatogonium. Those will turn into spermatids, and then they'll finally turn into spermatozoan. The plural forms of this are spermatogonia, spermatids, or spermatozoa. And if a spermatozoa finally enters the female tract, then it'll become fully matured with another process. And so the steps here below are stem cells, which are the spermatogonia. They will go through spermatogenesis to become spermatids. Then those spermatids will go through spermiogenesis to become spermatozoa. And then if sperm enters the female tract, they will finish going through another final process called capacitation, and that's where they'll become fully matured sperm. For females, singular, we start with oogonium. Those become polar bodies and oocytes. The polar bodies dissolve. The oocytes could be fertilized, and if they do get fertilized, then they turn into zygotes. Plural forms, oogonia, polar bodies, oocytes. The steps in oogonia could, those are the stem cells. Those go through oogenesis to become polar bodies and oocytes. And then if an oocyte gets fertilized by a sperm cell, then they go through fertilization and they finish becoming fully matured eggs. Okay, so like I said, that's probably a lot of information. You're like, oh my gosh, I kind of know what the words are that you just said. I just hope that these slides are going to be useful for you as you try to build the entire picture together. We have a lot to add to these slides, but for now, that's just where I kind of want to leave it. So my recommendation would be to take a sheet of paper and jot these down and leave like good chunks of space for section 1, section 2, and section 3, and then have ways to be able to come back to it and say, like, leave a little note for yourself next to females that says, like, oh, go look at the ovarian cycle here, or oh, go look at male GnRH pulses here. And hopefully that'll be helpful for you. If none of this is helpful for you, then you can just totally ignore this. We're going to talk about this all in detail anyway. I'm just trying to help organize all of the information. All right, so let's go ahead and start. So review. On the right-hand side, I have a view of a karyotype. And a karyotype is something that is taken whenever you're pregnant. They'll go ahead and take some of the genetic material from baby, and they'll map it out. So they'll take all of these chromosomes, and they'll put them in their pairs. They'll size them up, and usually 1 is the biggest chromosome, and then 22 is usually the smallest chromosome, and then X and Y, or X and X, or they have specific ways of being able to identify those from the rest. But then they pair them with each other. And the whole point of having a karyotype done is to tell the parents whether or not you might have extra or missing chromosomes. And then that can allow the parents to make decisions, or at least prepare for potentially whatever scenario it might be, that just to prepare. Here I have a male karyotype. So at the end, you'll see 22 pairs, and then the 23rd pair is X and Y. For females, this would be X and X. We call these sets sex chromosomes, and everything else are autologous chromosomes. So I'm going to come down here. I didn't want to make anyone print out these colors. I just think it looks prettier like this, so I just want to show it. But it's exactly the same thing. It's just another way of representing the male karyotype. And what I want to do is just kind of talk about nomenclature for a second, because everything kind of sounds the same. So we have chromatids. Chromatids are unfortunately also called chromosomes, not because of lack of knowledge, but sometimes it's just simpler to say chromosome instead of chromatids, especially whenever you're talking to the general public. So, but an actual chromatid is actually just one singular chromosome. If you put a chromatid next to its very similar chromatid, then we're going to call these sister chromatids. And the whole idea is that you get one from mom and one from dad. And whenever you're alive, a child, well, everything's fine and good, you might be using this chromatid in order to actually turn on those genes, or you might be using this chromatid to turn on those genes. And then that can be different for each pair. This is especially important whenever we talk about the sex chromosomes here at the end. Because if a male is male, they'll only use the Y chromosome for a short period of time, but then everything else falls on the X chromosome. And for females, when they have X or X, then they either use one X chromosome or the other X chromosome. And there's a lot of interesting genetic problems that can come from that. So that's why we highlight the importance in these intro classes. So we have 23 pairs of chromosomes. So a pair of chromosomes are a pair of sister chromatids, and we would call that one chromosome. Then we have one other term and one other term I want to mention. So all of these, let me go in yellow, all of these pairs, all the way down to 20, 21, and 22, all of these are autologous chromosomes. And so what that means is that as humans, we all likely have very, 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 very similar autologous chromosomes. My ones are going to look very similar to your ones, and so on and so forth. And that's part of, I shouldn't say that's what makes us human, but that is part of how you identify human versus non-human. And remember, all of these chromosomes and why it's colored like this is there's individual genes. So all these individual genes are like this, and they're all wrapped in really, really tightly. And we can actually see it with the naked eye. Well, if we zoom in a little bit, then we can actually see it with the naked eye. But remember, this is a lot of genetic material that needs to be organized in a really effective fashion, and all of these cells. So every single cell is going to have 22 of these autologous pairs, and at any given time might go into transcription and then eventually translation of just one of these chromosomes. So then we would have to take this out, move it out a little bit, make it not as condensed, and then go even further and make it even less condensed, finally get in the right enzymes like DNA polymerase, helicase, all of that stuff, and then finally be able to do transcription. The exception in humans is that I shouldn't say the exception, but the main difference between humans are these X and Ys. And these are, of course, specific to sex. So if you have XX, female, if you have XY, male. Right, yeah, XY, male. And these are interesting because usually the Y chromosome oops, sorry, that's the dog. Let me actually just close that. No idea why we're getting excited. I just said XY chromosomes and now we're getting excited. So the Y chromosome is super important because the Y chromosome really, I shouldn't say has one purpose, but it really does only have one purpose as far as we know. And that's just to turn on the rest of the genes to make male type features. Afterwards, the X chromosome really kicks in and does the rest. What else should we mention about this? Oh, okay. So the reason why I even bring any of this up is because I want you to have a little bit more of an appreciation of why we talk about mitosis and meiosis so in depth, and why do we ask you to know it so well backwards and forwards, is because this is a huge, important, fundamental piece of human biology that you need to understand in order to be able to apply really anything else whenever it comes to variances in the human population. So for here, I want us to think about mitosis. And in mitosis, we are going to replicate all of these cells, right? So somehow, let's say you damage a part, like you scrape your hand, or it's coronavirus season, so you're washing your hands really frequently and it's really dry, and so your skin has to rebuild itself a little faster than it normally does. So all those skin cells have to go through multiple rounds of mitosis. And now you don't want your new skin cells to be any different than your previous skin cells. You just want them to replace the old skin cells. So in order to do that, somehow we have to recreate this entire karyotype in each individual cell many times. So in order to do that, you're going to pull one chromosome, you're going to make a copy of that chromosome, and then you're going to hand it off to the new cell, right? And so we start with a mother cell, and then we end up with daughter cells. It's a little different in meiosis, because in meiosis, we're trying to make sex cells. And sex cells don't need to have as many chromosomes. They only need to have half as many chromosomes, or chromatids, because the intent is that they will have half, and then they will meet up with the opposite sex's half, and they will combine to make one new whole, and then that new whole will hopefully become a human being. So whenever we do that, we're not dividing these in half. Instead, we have to double them on both sides with extra chromosomes, and then we split them in half, and then we split them in half again. So we have a specialized name for that scenario, and that's over here in the lecture slides. That's one pair of homologous chromosomes, and whenever they're in a set of 4 like that, we call them a tetrad. Homologous chromosomes are 2 paired sister chromatids, so it's two sets of these next to each other. So it would be like if I put this right up next to this, but assuming that they're the same chromosome type. This will be important whenever we get to meiosis, so that's why I give you kind of this refresher on karyotypes and genetics. So let's talk about meiosis. So we start with, let me go in this color. So with mitosis, we start with a mother cell, and the mother cell has 2N. And all of these cells, and let me write mother cell, and all of these cells are going to be somatic cells. And what does somatic mean? Well, soma is the prefix. Soma means body. So these are all types of cells, every type of cell except sex cells. So everything, skin, hair, nails, bowels, eyelids, eyelashes, everything. And then these cells are replicating, and the intent of this process is to make multiple copies of the original cell. So in the same instance, we want to make sure that the next set of cells are slightly better than the previous set of cells. And so we're going to have some genetic variability where we cross over. And if you remember with crossing over, that's where we have one chromosome. Let me do different colors. One chromosome, and maybe it has this pink end here. And then we have the other chromosome that I should draw in the same color. So it's like this. And maybe it has like a yellow end over here. And during metaphase, maybe these two spots switch. So after anaphase, then you would see that this blue one has the yellow on its side, and the other one has the pink on this side. And the intent is that maybe the yellow works better with this set of genes rather than the pink with the previous set of genes, so that whenever we use this autologous chromosome, that maybe that would have a better advantage and it would make a better epithelial cell, for instance. We have to talk about anaphase. So anaphase is going to look different for mitosis and meiosis. So anaphase, we have separation of sister chromatids, and remember, sister chromatids are a pair of chromosomes. And then the results, 2 identical daughter cells with 2N each. So let's go ahead and draw this out. I know that you probably have done this in 111, so if you already feel comfortable with this, you can go ahead and fast forward. But for those of you who want a little bit of a refresher, let's go ahead and go through this. So the mother cell. So the mother cell is going to go through prophase. And in prophase, we're going to double everything. So 2N, so we'll still have N being the number of chromosomes present, and that's still doubled, so that we can eventually half that into 2 individual cells. So here, let's also draw some dots. So let me draw like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and here we'll do 1, 2, 3, 4, 5. What's that representing? That's just like the Golgi apparatus, the ER, the mitochondria, all of that stuff that we need to have in a successful livable cell. So I'm just going to draw them as dots. In the picture here in the slides, they actually give you like a better example of what's going on there, but I'm just going to do them as dots. And then we're going to have 2 different chromosomes to work with. Let's talk about a yellow one, and let's talk about like a green one. Okay, and since we just did prophase, now we're going to double them. So that's going to have its sister chromatid, and this one's going to have its sister chromatid. Perfect. Next step, or PMAT here, I can draw that PMAT so that would follow along. Next step is metaphase. In metaphase, we still have 2N. We still have all of those dots, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And all of those fancy chromosomes are going to line up down the middle. And then we have some, not centromeres, centrioles, and they're reaching out with their microtubule spindle fibers, looking for those chromosomes, and they're arranging them on the metaphase plate so that we can have an equal separation on both sides during anaphase, which is the next piece. Right, PMAT, yep, okay, anaphase. And here I'm just going to kind of draw it like this because they kind of separate, but they don't totally separate yet. And we have equal distribution, so 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And then that yellow goes over to this side, and the other yellow goes over to this side, and that green goes over to this side, and that green goes over to this side, and we still have those spindle fibers on both sides, and they start to not disintegrate, but the microtubules go back into the centrioles, and they wait for the next time that they have to do mitosis again. So then we go to telophase, and in telophase, and let's just do telophase and cytokinesis. Kinesis. Okay. And then that's when we actually get really, really close to separating the 2 cells, and cytokinesis is where we actually have that separation of the 2 membranes. This is done with a bunch of actin filaments. They all arrange themselves in a really interesting way at the plasma membrane in between those 2 cells, and they literally just cinch off each other, and then the plasma membrane reforms itself. It's really neat. And then now we have, I should have drawn 2N here, I apologize. And then now we finally have 2 daughter cells with our genetic material that we need. And we have the red 5 dots, 1, 2, 3, 4, 5, awesome. And so now we finished, and now we have 2 identical daughter cells. Perfect, and they're both 2N each, so these look exactly the same as what we started with. Okay, perfect. So just as a reminder, 2N we call diploid, and N we call haploid, as half. Okay, so let's talk a little bit about development for a second before we talk about meiosis. So in utero, you have when a mommy and a daddy love each other very, very much, right? Then they come together and they fertilize an egg. And that fertilized egg then becomes a zygote. And that zygote folds in on itself, replicates itself many, many times, and then you start getting a ball of cells. So that's what's represented here by zygote. And then a zygote becomes an embryo, and then an embryo becomes a fetus, and then a fetus eventually is a baby once delivered. So I drew an arrow in here between zygote and embryo so that we can show another picture. And in this zygote picture, you're just seeing one cell, and then it turns into the 8-cell stage, and then that 8-cell stage it becomes a morula, and then a blastula, and then has all these other specialized names. So if you're ever interested in this stuff, take a human development or a developmental bio class, and it'll go into all of these details. It's pretty neat. So once we're in a blastula, we have this like space in the middle. And if you remember from the digestive system, I mentioned deuterosomes, where we have to make a hole at some point, and that's the beginning of the digestive tract. And you can either start mouth first or anus first, and humans are deuterosomes, so we start anus first. And so that's what that hole is in the middle of the blastula. And then at some point, then that hole starts to fold in on itself in order to make other channels. I shouldn't say channels, other tubes in the human body. And right where I put that little star, that yellow star, is where the cells that are designated to become gametes are going to be. And they all congregate to that one area. And it all has fancy terms and all those fancy genetics that make it go there, and all these interesting cytokines and conversations between cells in order for it to get there. Super interesting stuff. But we're just going to call it the star. And that is where all of the gametes are going to go. And then everything else surrounding it in green are going to be the somatic cells. So those are what's going to turn into hair, skin, nails, bowels, and so on. And then those yellow cells are going to turn into the gametes. But this is super, super early on, super, super early on. And so we only have like 2 cells. And those 2 cells, that's not enough to start actually going through meiosis. Those 2 cells have to go through rounds of mitosis first, so they make duplicates of themselves, and then they go through meiosis. So we are going to focus on just those yellow cells from here on out because it's the reproduction chapter, and that all has to do with gametes. So males are going to, in utero, they're going to start with a couple. They'll go through different rounds of mitosis, or multiple rounds of mitosis, and they'll make spermatogonia. And remember, spermatogonia are the male sex stem cells. They will make a couple. They'll go through a couple rounds. They kind of have this like I shouldn't say mediocre, but like an even level of spermatogonia available to them until they hit puberty, and then this explodes. And they make multiple rounds, and they go through meiosis many, many times. Females, on the other hand, we start with a couple, and then we go through multiple rounds of mitosis, just like the males, to make oogonia, female stem cell sex cells. And before birth, we have about 7 million. So somewhere in between the embryonic and the fetal stage, we have about 7 million. Then once we're a fetus and we're going through the 3rd trimester, then we have about 2 million. Then we're born, then we become a child, then we go through puberty at about like 12. By the time we're between being born and by the time you actually hit puberty, you actually lose some of that 2 million, and you're left with about 400,000. And then those 400,000 are everything that you have. So those 400,000 are what you're going to use every single menstrual cycle. So every single menstrual cycle, you'll lose a couple. And this is, of course, going to happen every month. So this is why I mentioned this in the beginning, because it's really important to understand, again, why mitosis, why meiosis, why do we care so much? And I think this kind of helps lay out the why behind the what. Okay, so what is meiosis? Let's finally talk about meiosis. Okay, so with meiosis, and I'll draw a line, we're going to start with a mother cell, again, 2N. And we're going to start with spermatogonia or oogonia, or those stem cells. We're going to, again, have genetic variability, and we're going to have crossover again at metaphase. But meiosis actually has to go through 2 rounds. And what we'll see with the chromosomes is that the chromosomes have to double themselves more than what they did in mitosis. And then they're going to go through 2 rounds of divisions. And we're going to have meiosis 1 and meiosis 2. And that, therefore, we're going to have prophase 1, prophase 2, metaphase 1, metaphase 2, and so on. So we're going to have crossover happening twice, once in metaphase 1 and once in metaphase 2. During anaphase 1, we separate homologous chromosomes. That's different than what we talked about in mitosis. Then in anaphase 2, we separate sister chromatids. That's the same as what we talked about in mitosis. And then the final end result is we end up with 4 haploid daughter cells. They're all going to be 1N each. Okay, so let's go ahead and draw this out. What on earth are you talking about? So we start with a mother cell. These are going to be gametes. So they're not somatic cells. These are just sex cells, And let's say we have 5 dots to start with in the mother cell. And let's say we have those same chromosomes. So chromosome 1 and chromosome 2. Okay, so then we go through prophase 1. And you have to add the 1 afterwards. Also, please use this as Roman numerals, not oh my God, I think it's Arabic numbers. Is that what we call like this and this? Okay, so prophase 1. After prophase 1, then we're going to double everything, but we have to double everything a lot so that we have enough for those 4 haploid cells. So in order to do that, we're still going to be 2N, but those 5 dots, we want to make sure that every haploid cell is going to have those 5 dots. So those 5 dots have to multiply by 4, so we're going to have 20 dots. So 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And then those chromosomes, we're going to have enough of those chromosomes to satisfy having 4 different cells. So we're going to make tetrads. All right. Now we move into metaphase 1. And metaphase 1, we line up everything in the center just as before. So 2N, 20 dots, but I'm going to kind of put the 20 dots on both sides. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Chromosome, 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 chromosome. Cool. All right. Then they start to pull apart. Now we're going to pull apart during anaphase homologous chromosomes. So anaphase 1, and we go like this and like this, and these are still going to be 2N on both sides. And now we have the 10 dots here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I'm going to put the 2 yellow on this side, the 2 yellow on this side, and the 2 green on this side, and the 2 green on this side. Cool. All right. Then we do telophase and cytokinesis. So let's do this. Telophase 1 and cytokinesis. Cytokinesis, as a reminder, this is a process. This is not a step or a phase, so there's no such thing as cytokinesis 1 or cytokinesis 2. So cytokinesis is just the process of cinching off the cells. So we have telophase 1, and then we end up with, two 2N cells, and those yellow chromosomes rearrange themselves like this, and the blue ones rearrange themselves like this. Oh, and I should have drawn it on both sides like that. And then, of course, we have those 10 dots. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Cool. We've done meiosis 1. Awesome. Now let's go through meiosis 2. So this is going to happen on both sides of this. So each of these 2N cells are going to do the same thing. So now we'll go through meiosis 2. And this 2N cell is going to go through, but we're going to skip prophase. We don't need to do prophase again because we already doubled everything we needed to during the first prophase. So we're going to go straight to metaphase 2. And I'm going to separate everything equally and put my chromosomes down the middle along the metaphase plate. And now I'm going to put my 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Okay, then after metaphase 2 comes anaphase 2. And then we get our nice bubbles. And then we get N, 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 N. And chromosome, 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 chromosome. Da-doom, da-doom, da-doom. And then we get 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Then we do telophase, 2, and cytokinesis again. And then we get 1, 2, 3, 4, N, da doom, 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 and dots. 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Cool. Awesome. So now we have 4 haploid daughter cells. Awesome. And I want to remind you that this looks almost exactly like what we started with, except half the genetic material, because again, we want to meet up with either another female or male, male with female, female with male, sex cells to double that genetic material again so that we can start with a human being. Okay, so the reason why I draw it out is because it's really important to be able to draw it out so that you really understand it. It's one thing to look at these images in the book, and the book does a good job of describing what's going on. I think it introduces it at kind of a weird time in the chapter, so that's why I bring it up to the beginning of the chapter for us to talk about. I think it's 28-3, I included it on this slide, so that's where you can find this picture in the description. But it's always really important to find images that make sense to you and then make your own version of that image in order to really understand what's going on. One thing I want to mention, we talked about crossing over at metaphase 1 and metaphase 2, but I want to mention something else, and this is not something that you need to know, but I think it's super interesting and relevant, and it's something called non-disjunction. So non-disjunction is not necessarily a great thing. It's not normal, so let's draw out our tetrad, and normally this tetrad would go like this at anaphase 1, and then again through anaphase 2, and it'll separate it out. What non-disjunction looks like with the same chromosomes is instead of having an even separation between the two, instead you don't get an even separation and you just get 3 on one side and 1 on the other side, or during anaphase 2 you get 2 in one cell and nothing in the other cell. Now, for the most part, this doesn't, and it depends again on what cells this does this to. So if we have autologous cells, or autologous chromosomes that this happens to, we might end up with 1 extra chromosome. And typically, it doesn't necessarily create any problems, but it can. And usually, if it's a cell that is damaged, or if it's a cell and the chromosome that went through non-disjunction is a really vital chromosome, usually that cell will die off anyway and it won't actually be able to be fertilized in the first place because the cell has mechanisms to double check itself that it has the right amount of DNA at any given time. So the one example of autologous non-disjunction is Down syndrome. In Down syndrome, now I'm forgetting, chromosome 21, I believe, chromosome 21. So chromosome 21 is normally supposed to have 2, but in Downs we actually have an extra chromosome there. So Down syndrome is not necessarily going to ruin quality of life, but they do typically live a lot less, typically live a lot less. They typically live to mid-50s, mid-60s. They're unable to reproduce. Or well, I shouldn't say they're not unable to reproduce, but it is more likely that it'll be more difficult for them to reproduce than it would a healthy individual. They usually typically have cardiovascular issues as well, and some mental. I don't even want to say it's a. It is so they usually typically go through Alzheimer's and dementia at much higher rates than the normal population does. But these are very happy people. They're very energetic. They're very appreciative of life. They really enjoy anyone's company. So it's really interesting how 1 chromosome can have that much of a difference just in having it in excess. And like I said, if you have 1 cell that has 1 additional chromosome or 1 less chromosome, usually it doesn't usually make it out. So the reason why I mentioned non-disjunction is because in sex cells, this is different. So in sex cells, if we were talking about the same abnormality, we could actually end up with XXY people, or multiple X's. So there are multiple X's and one Y, or multiple X's and no Y. Oh gosh, excuse me. But it's really untypical to actually get XX and multiple Ys. And why is that? Because there's only one Y ever. So there would be no ability for Y to double itself and to give two of it into one cell. And if that doesn't make any sense to you, you need to go through meiosis a couple of times in order to really understand that. What's interesting is, for the most part, these people have actually no idea that they have extra X chromosomes. So for females, they will probably go throughout life never knowing that they have extra X chromosomes unless they had some sort of karyotyping done, or they had some infertility issues and they went in and they did all of the things that you have to go through in order to evaluate where the infertility may be. But usually these people are normal. And the reason why they're or why they experience a relatively normal life is because these X chromosomes, it doesn't matter how many X chromosomes you have, it will always only pick one and only work with that one. All the other ones get silenced and told, like, we don't need you, you're cool, it's fine, you just hang out. Males, on the other hand, depending on how many X chromosomes they might have, or how many extra X chromosomes they have, they typically do see some sort of dysfunction. Not always, it's not always, but there's a couple different instances. So if you have one extra X chromosome, it may, in every other cell, it might be activated differently. So this X chromosome might be activated in one cell, and in another cell, this X chromosome might be activated. And then this potentially changes the individual's experience with puberty. So sometimes they may go through puberty earlier, they might go through puberty later, they might develop more female characteristics than male characteristics, but they will also still develop the male characteristics. They might they might experience being female for most of their life until they hit puberty, and then testicles start to descend. So it's really interesting. There's lots of different diseases that come from having these extra X chromosomes and just one Y chromosome, but typically females with multiple X chromosomes do relatively well, and like I said, you don't really notice. So anyway, this is why I talk about non-disjunction. We will not test you on non-disjunction. I just think it's important and relevant and interesting, so that's why I share it. Let's continue. Nope. Okay. So in gametes, sex chromosomes, sex chromatids X and Y, after meiosis, you will receive this, this N cell we're talking about right here, you will receive either an X or a Y. So this N cell might have an X, this N cell might have a Y, this N cell might have an X, this N cell might have an X. And you're going to get one from mom, one from dad. So this X could have been from mom or dad. This Y was definitely from dad because mom doesn't have any Ys. This X could have been from mom or dad. This X could have been from mom or dad. Autologous chromosomes, after meiosis, you will receive one chromosome from either mom or dad, or one chromosome, any other chromosomes you'll receive from mom or dad. But it's unclear which one it is unless you went through extensive genetic testing. So over here on the right, I have the N cell, and I took that from this image over here, that N cell and its karyotype. So its karyotype looks really similar to this karyotype, but notice that it's in half. So we only have 23 half sets, or 23 chromatids. We're missing the sister chromatids. And if fertilization occurs, you can imagine that we have a female gamete over here, so with its X chromosome as the sex chromosome, and then the 22 other chromatids, and then that mixes with the male gametes set, and the male gamete is going to have 22 chromatids and one Y or one X chromatid, and then they combine, and then they will make a diploid cell, and then that diploid cell is the zygote, and that zygote can go on and continue through and become an embryo and eventually fetus, and so on and so forth. Okay, so I hope that all made sense. If it didn't, please go through and redraw it out a couple of times. At first, like I said, it can sound like, oh, okay, no, that makes sense, that's totally fine. But then if someone were to ask you to reproduce this, then all of a sudden it gets a little shaky. So I would practice explaining it to someone. That's honestly one of the best ways to really learn this. and think of scenarios of where things could go wrong. And next up, we'll do male anatomy, and then we'll finish up with female anatomy.